part of lecture 2, and in this part I'll talk about development of inocula for fermentation. So what is inocula or inoculum? Inoculum refers to living organisms such as bacteria or other microorganisms that is added to initiate a biological process. Okay, so like during fermentation, so what suppose you are doing a fermentation using microorganism. Okay, so that means you have to put microorganisms into the fermenter to start your process. However, you just cannot take the microorganism from your stock and add them into the fermenter. Okay, we cannot do that. So what we have to do first, we have to prepare a large amount of microorganisms which has certain characteristics which are in a specific physiological state. Okay, so we have to prepare those microbes first, then we add them into the fermenter. Okay, and this microbes or microorganism is called inoculum. Okay, that which we add to the fermenter. Okay, okay, and so we can define inoculum, the process of inoculum development like this: the preparation of microorganism from a stock culture to a state useful for inoculating a final productive stage is called inoculum development. Okay, so when we develop or prepare inoculum, it should have certain some desired characteristics. So these are those desired characteristics. The inoculum that must be in a healthy, active state, thus minimizing the length of the lag phase in the subsequent fermentation. I mean, if the inoculum are in their active stage means in exponential growth phase. Okay, in that case, there will be reduced lag phase in the final fermenter. Okay, and it must be available in sufficiently large volumes to provide an inoculum to optimum size. I mean, we cannot add less amount of inocula into the fermenter. Okay, so if we add very, insufficient amount, then it will, then the microorganisms need to grow first, they need to grow. Okay, so it will add some lag phase in the fermenter. Okay, then it must be in a suitable morphological form, it must be free of contamination, it must retain its product forming capabilities. Okay, so these are the desired characteristics that a good inocula should have. Okay, and when we, let's say we've already prepared the inocula, so when we add inocula into the fermenter, usually the quantity or amount of inocula used is between 3 to 10% of the medium volume. Okay, 3 to 10% of the medium volume of the final fermenter, and a relatively large inoculum volume is used to minimize the length of the lag phase and to generate the maximum biomass in as short a time as possible, thus increasing vessel productivity. Okay, so that means the inocula that we add in the fermenters, its volume should be between 3 to 10% of the total media volume, and we should add sufficient amount of, that means large amount of inocula into the fermenter. Okay, and the aim of inoculum development should be to obtain a genotypically identical copy of population that was stored. Increase biomass means we have to prepare a large amount of cells, right, to add into the fermenter. Then develop the culture to a physiological state suitable for the performance in the final production stage. Okay, so suppose, they have to be in the active form, actively growing form, otherwise there will be lag phase. Okay, so that means the cell should be in a particular physiological state, okay, which is suitable for the fermentation process. Okay. Okay. This figure, it shows the step. Shows the all the steps of a inoculum development process. Okay. So it starts from the uh, master strain. Master strain means, suppose you have the your strain in lyophilized form, or you stored it at liquid nitrogen. Okay, so usually we store the master strain uh, either in lyophilized form, or we keep it at my liquid nitrogen. Okay, so first we need to grow the master strain in like in a small culture, okay, in a small volume of media. Okay, so then, so usually, like for example, if you have stored it in liquid nitrogen, or in lyophilized condition, usually we use agar plate. Okay, and we used uh, uh, not selective media. Okay, basically we use English type culture, because as the cells are in lyophilized form or in very cold temperature, at minus 190 degree, 96 degrees Celsius, so definitely this strain needs some time to regain. Okay, to regain their capability to grow at fullest. Okay, so first we culture them in, usually culture them in enriched culture media, then we culture them in liquid broth. Okay, and we culture them in a way, or using that condition for that time, for that time, so they are in their maximum, so the maximum cells are in log phase. Okay, and we do this in usually we can use like very small amount. Okay, only few ml media. Okay, so then we transfer them in a bigger flask. Okay, so this can be, even 1 liter flask. Okay, you can use even like one 100 ml here, or even 1000 ml of media here. Okay, and again we culture this flask in a condition where maximum cell remains in log phase. Okay, so this is actually our step culture. Okay, so these steps, you can say that in these steps we prepare our stock culture. Okay, so once we uh, cultured our master strain to sufficient amount, then we aliquot them. Okay, aliquoting means we divide them in a small volume, such as you can put 20 ml in test tubes and store them at minus 20 degrees Celsius. Okay, don't put them back in liquid nitrogen. Just we can store them at minus 20 degrees Celsius. Okay, and this is called the stock cultures. Okay, so we have to do quality check of this stock culture. Okay, usually 3% of the sample we should check for purity and productivity. Okay, you should suppose you prepare 100 stock culture, so 3% means 3. From this 100 stock culture, you have to take randomly. You have to do this randomly. Okay, you have to take 3 test tube and check them whether they are pure, and also you have to check them their productivity. Okay, so whether they retain their original productivity. Okay, so then, okay, the next step, once you have the stock culture, then from this stock culture the inoculum development process begins. Okay, so first you have to take out one of your stock culture and culture them in liquid media. Okay, usually we do this in flask. Okay, because this volume is low. Your stock culture is only 20 ml. Okay, very low amount. So first you need to culture them in larger volume. Okay, so you can do this. You can culture them like in 1 liter or 2 liter. Okay, and then once you have cultured like for in 1 liter, then you have to put them into the seed fermenter. Okay, so seed fermenter means in this kind of fermenter we increase the biomass of our cell. That means we produce large volume of cells, and this seed culture are added into the final fermenter. Okay, so these are the steps involved in inoculum development process. Okay, this was the were the overall steps involved in inoculum development. Anyway, when we develop inocula for bacterial process, we should keep some points in our mind, and that is, okay. So the inocula for bacteria should be in their logarithmic growth phase and metabolically active. Otherwise, if we transfer the inocula, I mean, if the inocula is not active enough and we transfer them into the fermenter, the lag phase will be long. Okay, and a longer lag phase means loss. How? Because if we transfer the inocula in their lag phase, so this bacteria first they need to grow and be in the logarithmic phase. So it will require some time. So it will lengthen our fermentation process. And also in addition, and during this time, and when the bacteria will be in the lag phase, they will utilize the fermentation media for their growth. Okay, it is a complete wastage, right? Because if they were in their active form, active growth phase, that is logarithmic growth phase, they will utilize this media to produce the product, not for the growth. Okay. Okay. So not only the bacterial growth phase, I mean whether they're in lag phase or log phase, also the size and the physiological condition of the inocula can affect the length of lag phase. Okay, in the final fermenter, especially when we use sporulating bacteria. The age of inoculum is very important. Okay, so sporulating bacteria means the bacteria which produce spore. Okay, and the sporulation of the spore is usually induced at the end of the logarithmic phase. Okay, and if we use inocula which contain high percentage of spore, the lag phase will be very long in the fermentation process, because first the spores need to sporulate. Okay, then they will do the process, right? They will do the fermenting. So that's why age of inocula when we use sporulating bacteria is very important. Okay. Okay, so this flowchart, it shows the steps of inoculum development for production of vitamin B12 from Pseudomonas denitrificans. Okay, I mean, this bacteria Pseudomonas denitrificans is used to produce vitamin B12. Okay, and these are the steps of their inoculum development. Okay, so first, stock culture lyophilized with skim milk. We have to transfer the stock culture into the maintenance culture, and the maintenance culture is agar slop, which we incubate at 28 degrees Celsius for 4 days. Then once the maintenance is done, we have to put them or transfer them into the seed culture. Okay, so, and the seed culture for inoculum development of Pseudomonas denitrificans has two stages. First stage of seed culture is done in two DMQ flask, okay, containing 0.6 DMQ medium inoculated culture from one slop, okay, one slop, and incubated with shaking for 48 hours at 28 degrees Celsius. I mean, you just cannot put the seed culture, I mean this flask, at 28 degrees Celsius. Okay, you also have to shake them. Okay, there are shaking incubators. You don't have to manually do that. Okay, there are shaking incubators, so you have to use shaking incubator. This is actually very important. Okay, so if you don't use shaking incubator, you will not get enough inocula. So it's actually a must. You must have to use the shaking incubator. Okay, so this was the first step of stage of seed culture, and in the second stage, you need to use bigger flask. like 40 to 80 dmq. I mean, this is too much for a flask. So basically you have to use fermenter of this volume, 40 to 80 dmq. Okay, and in this fermenter you have to put 25 to 50 dmq medium, which is inoculated with 1 to 1.2% first stage seed culture, and then incubate them at 32 degrees Celsius for 25 to 30 hours. Okay, so the seed culture is prepared. Now you can transfer the seed culture into the production culture, that is your fermenter. Okay, and fermenter are very big. So like for example, for this one, for the pseudomonas denitrification nitrificans, and if we want to use them to produce vitamin B12, then usually fermenter of this volume 500 dmq is used. Okay, and in 500, and this fermenter, this is the volume of this fermenter is 500 dmq, and the volume of media is used is 300 dmq, and we have to inoculate 5% of this second stage seed culture into the media, and then incubate at 32 degrees Celsius for 140 to 160 hours. Okay, so then you will get the vitamin B12. I mean, after this you have to harvest the media and uh, I mean collect and purify your vitamin B12. Then this slide, I mean this flowchart, it shows the steps involved in development of inocula for anaerobic bacterial process. Well, actually, it shows the steps of a sporulating bacteria. I mean, the inoculum preparation using a spore. Okay, so I mean, we can actually also use a spore to prepare inoculum. So how we can do that? So first, heat shock the spore. Okay, so and heat shock the spore suspension and inoculate into 150 cmq of potato glucose media. Okay, I mean actually what you have to do, first you have to suspend your spore in into potato glucose media. Okay, and the volume of the media you should use is 150 cmq. Okay, first make the suspension, then heat shock, and then do heat shocking. Okay, so after that what you have to do, you have to culture used as inoculum for 500 cmq molasses medium. Okay, I mean first you heat shock them at 150 cmq volume, then you have to transfer them into 500 cmq molasses medium. Okay, and then you have to transfer them into bigger volume 9 dmq molasses medium. Okay, and then you have to transfer them into your final fermenter. This is actually I mean until this stage you have prepared the seed culture, so now you transfer this seed culture into your final fermenter. Okay, which is usually very big one. Okay, so please notice one thing, that the media for this fermentation is molasses. Okay, but when we sporulate the spore, okay, or when we germinate the spore, glucose medium was used. Okay, so this is actually normal. Sometimes I mean we have to use a different media to facilitate the proper germination. Okay, okay, and one thing, I mean the heat shocking. I mean, why we need to heat shock the spore? Heat shock stimulate germination of the spore. Okay, and also in addition it removes the weaker spores. Okay, so if the spores are weak, they will not germinate properly. Okay, so they will be just cause some problem in the fermenter. Okay, there will be wastage. So that's why we need to stimulate the weak spores. Okay, so we can do this by uh, heat shocking. Okay, and heat shock also stimulate spore germination. Okay, and the production stays inoculated with a very low volume. Okay, okay, so that's all was about the inoculum development. Thank you.